Hello, fellow biologists. We are now in section 11.2 of Unit 3, Segment 3. So this is the second lecture video in a 5-video series. Now we are going to cover cell cycle and mitotic cell division. And just to note that the cell cycle material actually is derived from the last part of section 11.1. But I have merged it here with mitotic cell division because it focused more on cell cycle in eukaryotic cells, where mitotic cell division also take place. Your reading assignment for section 11.2 is found in pages 227 and through 231 of your textbook. So the learning objectives here are plentiful, but the main objective here is to describe the processes that occur during cell cycle and mitotic cell division. And it is worthwhile, in fact, to go through each of the sub-objectives because that will help you focus your listening to the important points relevant to each of these objectives. So this would be serves as a guidepost for you because this is a very lengthy video. All right. So 11.28 learning objective is to describe the two fundamental processes in mitotic cell division in eukaryotes. This will touch upon the two basic stages of the cell cycle, as we will see. 11.2b is going to describe the events that occur in the various stages of the cell cycle. 2c would be to explain the conditions that result in cells entering a special case or a special condition called G0 phase. D, explain how genomic DNA is packaged into chromosomes in eukaryotic cells. It is important for us to understand how chromosomes are packaged because they are critical in the mechanical distribution of the genetic material during mitosis. 5, or E, describe the number of chromosomes and their naming conventions in human cells. We need to start building a vocabulary of descriptions of chromosomes, and that is because this will be a important foundational background for you to prepare yourself in DNA replication as well as in genetic transmission of traits when you touch upon Mendelian genetics. So we will have to try to link the DNA that you're learning in DNA replication topic with the genes that are transmitted from one generation to another. That is the basis of Mendelian transmission. F is going to be for you to differentiate between what we call as homologous chromosomes and sister chromatids. Again, this is a vocabulary that you need to not just memorize, but need to understand where they are in the course of mitosis. G, to describe the chromosomal contributions made by each parent during fertilization. So the process of mitosis and the cell cycle is, of course, coupled with the event of fertilization in order to maintain the constant number of chromosomes from one generation to another. And so fertilization is therefore an important aspect of the concept that we need to learn in this section. You also need to be able to explain how replicated chromosomes remain paired after S phase. You will see why this is important later on. Describe the events that occur during the stages of mitosis. And finally, to differentiate between the process of cytokinesis in animal and plant cells. So for the self-assessment questions, in order to establish your understanding of this section, please try to, again, answer these following 5 questions. First, which DNA sequences are more alike? A pair of sister chromatids or a pair of homologous chromosomes? 2, what are the three situations in which mitotic cell division occurs? 3, what are the 5 stages of mitosis? Draw the structure and position of the chromosomes at each stage. So you can already sense that in these particular questions, we are focusing on um, the DNA and the behavior of the chromosomes during the process of mitosis. 6, what would be the consequence if a cell underwent mitosis but not cytokinesis? And 7, how does cytokinesis differ between animal and plant cells? So let's consider for a moment why the mitotic cell division in eukaryotes is more complex than the simple binary fission that we encountered earlier for prokaryotes in section 1.1. First of all, eukaryotic cells reproduce by mitotic cell division because certain elements are quite challenging for the eukaryotic cell to be able to distribute their genetic material to daughter cells. And that is, genome is large and linear, as I've already alluded to in section 11.1, whereas in prokaryotes, the genome is small and circular. And another important thing to consider, and which causes a large mechanical challenge, is the fact that DNA of eukaryotes is found in the nucleus, whereas in prokaryotes, the DNA is found in the cytoplasm. So the core concept that I hope that you will get from this section, and I would like to repeat, is that mitotic cell division is the basis of asexual reproduction in unicellular eukaryotes. For example, in yeasts, for example, the means by which they propagate themselves or they multiply themselves with these unicellular yeast cells is simply by simple mitotic cell division. But it also is the process by which cell divides in multicellular eukaryotes. And I would like to point out something very important here, in that the part of our bodies as multicellular organisms that undergo mitosis are our body tissues, excluding the special sex cells that produce eggs and for in female and sperm in humans, which as we will see in the next section is generated. The sex cells are generated by a process called meiosis. Okay, so the rest of the cells in our bodies, ranging from our brain cells to liver cells to skin cells, and especially our skin cells, which divide a lot. All of these, called body cells or somatic cells, let me write it down. Okay, they all undergo the process of cell division involving mitosis. And mitosis, in fact, that term is strictly speaking does not refer to cell division in general, but it refers to the division of the nucleus or nuclear division. Okay, but coupled with the process of cytokinesis becomes the cell division in eukaryotes. And that's why we say that it's the mitotic cell division is more complicated than in prokaryotes, because of these two fundamental differences that we see between eukaryotes and prokaryotes. The process of mitosis is simply a small part of a large and complex sequence of events that happen on a very regular cyclical basis called cell cycle in eukaryotes. Cell division in eukaryotes proceeds through a number of sequential steps that make up what we call as the cell cycle. And the cell cycle consists of two distinct stages. We have the M phase, and the M phase consists of mitosis and cytokinesis. Okay, and then um, the in just to say a brief word about M phase is that the time during which the parent cell divides into two daughter cells in this particular stage vary in length, but it is relatively short in within the context of the entire cell cycle. So it can go from a few minutes to an hour in order to take place. But the interface, which is the whole sequence here that intervenes between two successive M phases, is called the interface. So the interface is simply defined as the time between two successive M phases. And as we will see, they actually consist of a whole lot of integrated and complex events that are even finely regulated in order to assure that normal M phase happens during each cell cycle. So let's have a closer look at interphase. Interphase lasts much longer, at least 10 times longer than the M phase, about 10 to 14 hours. And during this stage, the cell makes many preparations for cell division to take place during M phase. And these preparations include two basic types of preparations. First is to prepare all of the machinery that is required for the replication of the DNA in the nucleus, and B, to increase in the cell size or mass so that each daughter cell receives sufficient amount of cytoplasmic and membrane components. So in the interface, therefore, although visibly nothing may be happening if you observe it under the microscope, but biochemically at the molecular level, there is so much activity going on to make all of the preparations that are necessary to replicate the DNA and to, in fact, replicate or to produce more of the organelles and infrastructure at least twice as much as the original cell in order to prepare it for the next cell division. So in other words, we can consider the interface as, in general, a series of growth phases. So what are these phases of the interface? So it can be subdivided functionally into four phases. The first one is the G1 phase. And in the G1 phase, or sometimes called the first gap phase, the size and protein content of the cell increases. So there is much growth happening here. So the cell content, there's an increase in mass. Again, as I said, organelles are being synthesized here. And moreover, there are many regulatory proteins are also made in this stage and activated, like kinases and phosphatases, which we already encountered in chapter 5 on cell signaling. And in fact, these kinases and phosphatases are regulatory proteins that are part of an exquisite signaling pathway that we will later on see in, I believe, in section 11.4 okay, to be involved in making sure or in monitoring the stages to make sure that each stage is actually doing its job in preparing the next stage. So in this particular case, the G1 phase is a preparatory phase for the next stage, which is the S phase. So all of the materials that are needed for the synthesis of DNA must all be manufactured or produced during the G1 phase. Okay, so the S phase then is called the synthesis phase, and this is a very, very special phase because this is where the entire DNA content, the entire genome of the nucleus has to be replicated and or duplicated, and has to be duplicated faithfully. And so if something does not happen correctly here, of course, this would be very unhealthy to the cell. It causes harm, and therefore it is therefore significant that the replication process here must be done in its entirety and in a very faithful way. Okay, so this is where DNA replication takes place. And this then is followed by the G2 phase. The G2 phase, or so-called the second gap phase, this is the phase where the cell prepares for mitosis and cytokinesis in the M phase. So this is a place where more proteins are getting manufactured and other materials that are particularly required for the M phase, like microtubule proteins, all of the mitotic apparatus, and even other regulatory, regulatory proteins that ensure that the M phase is happening correctly. They are all getting prepared at G2 phase. Okay, and once G2 phase is completed, then of course we now go to M phase. But in certain special kinds of cells, especially those that are approaching maturity or differentiation phase, there is a very special phase called G0 phase. Okay, and here the cells will stop dividing, but they will continue to perform their functions. So brain cells, for example, can stop dividing and but still continue to do their work. So although initially people think that brain cells can no longer divide, but there are evidence that brain cells can still divide, but at a very super slow frequency. So most of the time they enter into what we call as the G0 phase. Kidney cells, for example, also do this. So what we can say, therefore, is that the cell cycle, the rate by which the cycle happens, really differ in the types of tissues that are in the body. So for the case of skin cells, usually within 12 hours, the skin cells of your body gets replenished. So essentially, when you wake up in the morning, you're actually a completely different person from the evening before, because most of your epidermal cells have already been replaced. Okay, so something like that. So it's very intriguing, really. And then of course for some cells, there are special types of cells where you actually don't have an interface. In other words, cells simply keep dividing themselves. There are certain cases in insects, for example, where this happens. So there are all kinds of variations in the cell cycle. So what we're seeing here is just an average or generalized schematics, but you can actually observe different kinds of variations, both in the length of the G phase of the interface and the M phase, as well as whether interface happens at all. So having looked carefully at the different stages of interface, we are now poised to consider the events that are happening in the M phase. That includes mitosis and cytokinesis. But before we enter into that discussion, we still need to have a foundational understanding of the nature of the DNA as it passes through the M phase. The DNA is actually highly organized inside the cell. If you have a single DNA for every chromosome, and if you unwind it, it will actually be about an average of at least 2 inches for every chromosome that is inside the cell, if you unwind it completely. And so it means to say that the length of the DNA for each of the chromosome that needs to be distributed evenly, okay, in during mitosis, has to be packaged in a very tight space, at least a 10 million fold or 100 million fold, because the cells are super tiny, right? They are microns in diameter, and so the nucleus is even smaller than that. So in eukaryotic cells, therefore, the DNA is organized with special type of proteins called histones and other proteins as well into what we call as a chromatin. So this is like a beads on a string structure where you have histone proteins and DNAs wrap around them. Okay, so this is your chromatin, and your chromatin will then has to compact itself even more, fold back, and pack itself even more into multiple layers or degrees of compaction to form what we call as the chromosomes, which then become visible under the microscope when you use some special stain. And so here what we have in this figure is actually a stain of all the different types of chromosomes that you find in humans, okay? And here what is illustrated are homologous chromosomes, or meaning chromosomes that are not nearly not identical, but they are similar to each other because they have possess the same sets of genes in each one of them. Okay, and so these chromosomes are called homologous chromosomes. So individuals that have homologous chromosomes are called diploid. So humans are therefore diploid organisms because we have homologous chromosomes in our cells. In other words, the other way of defining diploid cells would be cells that have two sets of chromosomes. Okay, and so in this particular case, most cells of the human body contains 46 chromosomes or 23 pairs of homologous chromosomes. And what you notice here is, of course, a display based upon the staining pattern of the chromosomes. And when you have a display like this for geneticists, this is very useful because it allows them to identify all the 23 pairs of different pairs of chromosomes in the cell, in each cell of human being. And this kind of a display is called a karyotype. And the different colorations here is because of the different staining dyes that are used to color the chromosomes. And you can see that each homologous chromosome or each pair of chromosome are very different from each other, both in the pattern of the staining, but also in the positioning of what we call as the centromere. Okay, and we will later on describe what that is. Okay, and this is a very informative way of looking at the different chromosomes, and the reason is because there are many, as you will see later on in your genetics topic, that there will be a lot of abnormalities with chromosomes in a human genome that cause human diseases. And they can be visible, these types of abnormalities can be visible in displays called karyotypes. Okay? The human karyotype here shows 22 pairs of what we call as homologous chromosomes that are numbered 1 to 22 from the longest to the shortest. So they all appear to be very different in size. Okay, some are large, some are short. And of course, a very special pair called the sex chromosomes. Okay, and in female, there are two what we call as X chromosomes, and in male there is one X chromosome and one Y chromosome. And going back to the definition of the homologous chromosomes, as I already indicated earlier, homologous chromosomes carry the same set of genes. And how is that? How has that come about? Because one set came from the mother, and one set came from the father during the process of fertilization. Okay, so now you understand the basic principle of the DNA organization, and it is important to highlight this because you need to have an appreciation that the greatest challenge, in fact, in mitosis is to be able to organize your DNA in order and replicate them and make sure that they are rendered capable of being transported or moved around in a very precise manner during the process of mitosis. And so condensation of this chromosome into a highly condensed form that can even be visible under the microscope is a critical mechanical solution for ensuring that chromosomes can be distributed in a precise and safe manner. Because you can imagine if these are all unwound, it is really difficult for all of these DNA molecules to move around because they can easily be entangled and broken apart. There are two more terms that we need to be familiar with in terms of the structure that they represent in order for us to fully understand the mechanics of mitosis. And that is the term centromeres, which I introduced to you in the previous slide, and the term chromatids. Okay. So here on the left side is a representation of what we call as a homologous chromosome. And in this case, before S phase, homologous chromosomes are unreplicated. Okay, and they exist as in a loose form, as we have seen already in the form of a chromatin. Okay, and in the chromatin state, which is the loose form of the chromosome, an unreplicated chromatin before S phase, okay, which is this left side here. Whenever you see a figure in your textbook of a single-stranded chromosome, it represents a single double helix, like so. So each one of them is made up of a linear DNA, and that DNA is always double-stranded. It is not single-strand. So students always have have a misconception that when you say DNA, they can or chromosomes, they think that these chromosomes are separate strands of DNA, but actually they are, both of them are double-stranded DNA, and that they are homologous, meaning that they contain the same set of genes. Okay, and every chromosome, okay, has a very special structure called centromere. This is a structure that holds the two arms of the chromosomes. Okay, and they can either be positioned in the middle, or sometimes you find them in somewhere asymmetrical, closer to one arm, end of the one arm or the other, or sometimes you can even see them at the extreme end. Okay, but it is the centromeres that hold the two arms of the chromosomes together. Okay, and holds the DNA. Now think, of course, that this DNA is not naked DNA, but they are all complex with histone proteins. Okay. Now during S phase, what happens is that this DNA, each one of this DNA in each of the homologous chromosomes gets replicated, and that of course includes the DNA in the centromere. This because the centromeres are not just made up of proteins; they are also special sequences of DNA that the proteins bind to in order to make the centromeres. So a full DNA replication during S phase at the interphase stage will then cause a formation of a double-stranded chromosome, like so. Okay? And each of these double strand is a perfect duplicate of each other. Okay? So the sister chromatids are identical, or has to be identical, because it is generated by DNA replication. Although each homologous chromosomes are not exactly identical to each other, the sister chromatids are identical to each other. Okay? So in all sequences along the way. And they are called sister chromatids, by the way, because they share a single centromere. And so what is important that I need to emphasize here early on is that if you want to count the number of chromosomes inside a cell, the way to do it is to count the number of centromeres that is holding the strands. So it is not the number of strands that you see that is the basis for the number of chromosomes, but it is actually the centromeres that hold those strands. So the total number of chromosomes is actually equal to the total number of centromeres that you see in the cell. So this is the key, the total number of centromeres. And just to repeat myself again, it's nice to repeat concepts. A cell with one complete set of chromosomes is a haploid, but a cell with two complete sets is diploid, which means that they carry homologous pairs of chromosomes, just like as we saw in the previous slides. So the human body cells are diploid because they contain 23 homologous pairs of chromosomes in females. Okay, but in males, you have an X and Y. The X and Y are strictly not homologous because one is a large chromosome, the other is actually a tiny chromosome. There is only a partial homology between them. But it's something that you can discuss in your genetics portion. Okay, so this is the concept of centromeres and the concept of chromatids. Okay, so now we are ready to look at M phase involving mitosis and cytokinesis. So mitosis takes place in 5 stages, each of which is easily identified by events that can be observed under the microscope. So when you look at cells in interphase, you cannot distinguish specific chromosomes because they are all long and thin, because they are in the form of what we call as a chromatin. Okay, but as the cell moves from G2 to the start of mitosis, which is our nuclear division, then the chromosomes become condensed and they become visible. And therefore, we can examine them under the microscope. And as I said, we can identify identify 5 stages of mitosis under the microscope. So here we go. The first stage of mitosis is called prophase. And prophase is characterized by the gradual appearance of visible chromosomes as shown here. So these blue things that you see here under the microscope are actually chromosomes that are beginning to appear. They are called at this point chromatin fibers. Okay, and what is not shown or clearly shown in this actual figure is the nuclear envelope. So the nuclear envelope is still intact in the stage of prophase, and there is also a special organelle called the centrosome over here that gets duplicated, and they start to move to opposite poles. And while they move to the opposite poles, they generate a special string-like structures called mitotic spindle. And these mitotic spindle are simply made up of proteins called, and remember this, microtubules. Okay, and these are very important because it is microtubules that will pull the chromosomes apart into separate daughter cells, as we will see shortly. Now, centrosomes are what we call as microtubule organizing centers in animal cells. Okay. Now, every cell requires a microtubule organizing center. And in animal cells, this is called centrosomes, and they are these ones that are migrating at opposite poles during prophase. Now, in plant cells, they also have microtubule-based mitotic spindles, but they lack these centrosomes. The next stage is called the prometaphase. So during prometaphase, the nuclear membrane completely breaks down. So there's no longer any distinct nuclear region. And the microtubules of the mitotic spindle, as shown here in this figure here, in green, okay, attaches to the chromosome. So this is clearly shown in this figure here, and it's in these schematics. You will see individual chromosomes with some microtubules attached to their centromere. So actually, the microtubules are actually very dynamic during this stage. The microtubules would grow and shrink to explore the region that was once the nucleus. Now, you notice here that the chromosomes are highly condensed at this stage. Now, as the ends of the microtubules encounter the chromosomes, they attach to the chromosomes at their centromeres and start moving them towards the middle region of the cell. Now, let's take a closer look at the events that are happening in the centromere while the chromosomes are being moved around during the process of mitosis. So this is a schematics of the chromosome that has, of course, at this stage during mitosis is in the sister chromatid stage. In other words, the chromosome is double-stranded, and which is held by the centromere. Now, two protein complexes called kinetochores, okay, they are associated with the centromeres. Centromeres here is here, okay. One on each side of the centromere, colored in green. So there are two complexes of kinetochores that are associated with the centromeres on both sides, and they form the site of attachment for the spindle microtubules. Okay, this arrangement ensures that each sister chromatid is attached to a microtubule radiating from one pole of the cell. So, and these arrangements is important because it sets up the next stage of mitosis, which is the metaphase stage. So metaphase is one of the most visually arresting, visually distinctive stages of the cell during mitosis, as shown actually in this image. So you see the beautiful linear array of chromosomes that are stained in blue, and the spindle microtubules in green as they try to align them together in the middle part of the cell. So during this stage, the chromosomes are therefore being lined up in a single plane that is roughly equidistant from both of the what we call as the spindle poles as represented in these schematics. So this is metaphase. And the reason why this happens is becomes very evident in the next stage. So the next stage is anaphase, and here a most remarkable event happens. Okay, so during this anaphase, the sister chromatids separate. Okay, and the centromere holding this pair of sister chromatids together divides, allowing therefore the two sister chromatids to separate from each other. And now each can be called a chromosome because they now possess their own very centromere. And so this is the reason why an alignment has to take place in metaphase so that it ensures that the sister chromatids would split apart from each other and move on separate poles, okay, in the dividing cell, in ensuring that there is an equal copy of each individual sister chromatid being distributed to each side of the cell during anaphase. So it's really a beautiful design, a simple logical arrangement happening during metaphase, allowing for the equal distribution of all sister chromatids through the splitting or the division of their shared centromeres, allowing them therefore to become now individual but identical chromosomes on each side of the cell. Beautiful. And now we come to the last stage of mitosis called telophase. Now, during telophase, a complete set of chromosomes arrives at the spindle pole, and cytosolic changes occur in preparation for the cell's division. The microtubules of the mitotic spindle break down here and they disappear. So you can see them here in the schematics or in the image slowly fading away. And the nuclear envelope actually begins to reform around each other, not visible in the image, but is actually happening and depicted here in the schematics. Okay, and the nuclear envelope, as they reform around each set of chromosomes, they create two new nuclei. At this particular stage, you still have the same cell, or one single cell, but they are now about to split up. Okay, but they have not completely divided yet. That is called the cytoplasmic division that will happen after the telophase stage. Once the nuclear envelope is reformed, the chromosomes now decondense, okay, and become less visible. And this stage marks the end of mitosis. So after mitosis, which is the division of the nucleus, comes now the division of the cytoplasm, or called the cytokinesis. So there are two distinct modes of cytokinesis in animals and in plants. So in animals, what we have is that as mitosis ends, cytokinesis begins and the parent cell divides into two daughter cells by the formation of a ring of actin filaments. So actin is a protein, and it is a filamentous type of proteins, like a string, and they can actually form contractile rings, okay, that very reminiscent of the FTSZ ring that you see in bacterial cell that we saw in the early, previous section. And so this contractile ring forms against the inner face of the cell membrane at the equator of the cell, as shown here in this image, that is perpendicular to the axis of what was formerly the spindle. Now this ring contracts, so there is contraction happening, very reminiscent to what happens with actin in muscle cells. Okay, and as it contracts, it pinches the cytoplasm of the cell, and eventually dividing the cell into two. So this is how the cytoplasmic division happens in order to complete the process of cell division. So first nuclear division by mitosis, and then cytoplasmic division by cytokinesis. And this is how it's done in animals. Now, cytokinesis in plant, as I have indicated earlier, is very different from animals. Since plant cells have a cell wall, the division of the cell is achieved by constructing a new cell wall. Now, during telophase, the plant cells form a structure called the phragmoplast. It is not shown here, but it is a term that you might want to be familiar with. Phragmoplast. Okay, and that phragmoplast forms in the middle of the cell. So instead of forming a constriction outside, the cytokinesis actually starts in the middle part of the cell. Okay, in the middle of the cell. The phragmoplast consists of overlapping microtubules. So instead of actin, we actually have microtubules, okay, that guide vesicles containing cell wall components to the middle of the cell. So new cell wall materials are being built by microtubules delivering vesicles into that area to deliver cell wall components. And during late anaphase and telophase, these vesicles fuse to form a new cell wall. So that is new cell wall is now what we call a cell plate, as indicated in the image over here. Okay, and this cell wall, the cell plates in the middle of the dividing cell, once it is large enough, it fuses with the original cell wall right there, okay, at the perimeter of the cell, and that ends or completes the process of cytokinesis in plants. So that completes section 2, folks. Please, again, take time to answer these Riff questions. There are two of them in this section. I will quickly read them for you. For one, colchicine is a drug that blocks the assembly of microtubules. If dividing cells are treated with colchicine, at what stage of mitosis would you predict the arrest would occur? A, prophase, B, metaphase, late anaphase, D, telophase, and E, G1 of interphase. Number 2, a skin cell in G2 of the interphase has blank as much DNA as it had in G1. A, half, B, twice, C, exactly, D, 1/4, or E, 4 times. Okay, so after answering this Riff question, I would expect you to go and proceed to lecture video section 11.3. Thanks.